Okay, so today I want to spend some time on revisiting or examining some of the assumptions that we commonly make in a lot of when we learn about basic population genetics, of which all of you now have a fair idea of a wide range of sort of core concepts and work that has happened in population genetics. There's a lot more, of course. It's not possible to cover, all the breadth of and the depth of the work that has happened in this field in the last 50 years or so. But I want to focus on a few specific sets of assumptions. So like I mentioned last time, identifying sites under selection is a super important aspect of what we do in population genetics. A lot of population genetic analyses and evolutionary analyses are based on identifying sites under selection in the genome. And I realized that I forgot to talk about this one very important case of DN-DS ratios, which you'll see very, very frequently being used in a lot of work. So what is this DN by DS ratio? Simply put, it's talking about the number of non-synonymous substitutions that you observe in a population at a particular set of loci, for example, relative to the proportion to the number of synonymous mutations or substitutions you observe. And of course, normalized by how many of each of these categories of mutations are possible given the sequence that you have, right? So you're basically saying, let's count up the number of non-synonymous substitutions we see. Let's imagine you're doing this analysis across different species, different taxa in some clade of interest. So for example, we can think of analysis of homologous genes of humans and a bunch of other primates. And we're interested in asking what kind of selection or identifying sites in the human lineage which may have evolved under selection compared to the other primate lineages, right? So what we would do is in this phylogeny that we have in front of us, we would sample different lineages. So we might sample different species, humans, chimpanzees, gorillas, other apes, and so on and so on. And for each of them, we've sequenced some homologous set of sequences. And we're now comparing all of these, and we're making first a best estimate of what was the common ancestral sequence of all of this group of taxa. What did that look like? What was that sequence? And for that, we'll compare with, let's imagine, other mammals, right, which are not primates, to get an idea of what was the likely or the most parsimonious sequence of our shared common ancestor of all primates. And then we can say, okay, relative to that, how many or what kind of substitutions are we seeing in each lineage of extant species that we have access to right now, right? And in that comparison, we can then say how many of the substitutions are non-synonymous, how many are synonymous, and relative to how many of them could have been non-synonymous and how many would have been synonymous, right? In general, you have fewer possible synonymous substitution given the nature of genetic code. So if you remember, we spoke yesterday, there are triplets, and the third base is usually the one which allows for movement across different synonymous codons, and these mutations are synonymous. A subset of second base mutations are also synonymous, okay? But overall, about 2/3 of the mutations are going to be non-synonymous, and 1/3 of mutations are going to be synonymous, slightly more than 1/3, all right? So given this 2/3 and 1/3 more or less distribution for any given sequence, we can calculate what is the expected DN by DS ratio if sites are evolving under complete neutrality, right? And so a DN-DS ratio of more than 1 indicates positive selection for non-synonymous substitutions. And why is that the case? We're basically saying, compared to how many non-synonymous substitutions were possible in this sequence, are we seeing many more non-synonymous substitutions compared to the relative increase in the rate of synonymous substitutions? Now, remember, why are we taking this ratio, right? We're taking this ratio because we're assuming that synonymous mutations are evolving neutrally. So that's our neutral baseline. And it's a way that we can estimate some kind of local mutation rate, right? So for example, we know now that mutation rates locally will vary across different parts of our genome. So the DN by DS ratio seems like a really great way to estimate what is happening so that we account for all these differences across different parts of the genome. And for the same homologous gene, we can compare across species, for example, relatively easily, saying in general in this locus is the number of non-synonymous substitutions that we observe in the human lineage much higher than what we have observed in other primates. And if that is the case, we might make a prediction that this locus is therefore evolving under positive selection in the human lineage, because we see many more non-synonymous substitutions which are likely to have or more likely to have functional impacts. All right. And the flip side of this is that a dn by ds ratio of less than 1 indicates purifying selection against non-synonymous substitutions. So what we're saying there is in the human lineage, if we're seeing many fewer non-synonymous substitutions than expected by chance, then we are saying that maybe it's because selection has thrown out or led to the purging of these non-synonymous substitutions. Okay. Now, of course, all of this hinges on synonymous sites evolving neutrally. And as I showed you earlier, there are some issues with this assumption that it's not a great assumption always. And the bigger problem we're facing at the moment is we don't know how often this assumption is violated, under what conditions, in which organisms, how frequently, and so on. So does this mean that we should never assume that synonymous sites are under selection? Based on what we have seen, in general, it seems to be yes. But of course, we don't have enough data from enough number of species to be able to firmly understand this. I showed you some information on the distribution of fitness effects of synonymous versus non-synonymous sites in studies with microbial evolution typically. And there it seems like there is a weaker impact in general of synonymous sites compared to non-synonymous sites, but the distributions look very similar, right? So on the whole, many times it is probably a fair or reasonably fair assumption to say that synonymous sites are evolving neutrally, or are certainly evolving more neutrally or under weaker selection than non-synonymous sites, but it's not always a good estimate, okay? And I'll show you some examples of how sort of assuming blanket that synonymous sites are under selection is going to be a problem. Any questions at this point about DNDS ratios? Okay, all right. Shubha?
Deepa, I just had this question. When you look at DN by DS ratios, you look at these ratios across different species, right? For the same homologous region of the gene.
Yeah.
Okay.
Yeah. So that's the original intended use of DNDS sequences, because what happens is that effectively across these longer time scales where lineages have diverged across millions of years, you expect that these ratios are a good indicator of basically enough mutations and substitutions would have accumulated to give you a good estimation of selection versus neutral evolution. But people also use them to identify sites under selection, sort of comparing across different parts of the genome. And that is relatively a little bit more problematic. So there was a paper a few years ago from Sergei Krajimsky and Joshua Plotkin showing that within populations or within lineages, if you look at comparisons across different groups of populations or different parts of the genome, the assumptions that go into saying that DN by DS is a good indicator of relative amounts of selection doesn't hold up so well. It's not as sensitive to differences in selection strength. So I think they did simulations and they did some theoretical work to show that. So within populations, it's not as great an indicator. So basically across two loci, saying which loci are under stronger selection. It probably still works if there are very large differences in the nature of selection or the strength of selection in two loci, but it's not as sensitive.
Okay, thank you.
Any other questions? Okay, good.
I have a question.
Yeah.
Like, can we look into the selection process or selection pressure in the case of a gene family also, rather than species? Gene family, is that possible?
What do you mean gene families?
Gene family in the sense, for example...
I know what are gene families, but...
I know what I know. If there is kind of duplication event and there are a lot of analogs, so we look into selection in that case.
So it would be similar to the example I gave, right? Where you're looking at two different loci and you're asking which one is under stronger selection compared to the other. So the same issues apply. So it's not as sensitive. Of course, if the differences are huge, so for example, imagine one paralog has been completely pseudogenized, right? So now it should be evolving under pretty much under drift because it has no function and it is not under selection for its function. Then another copy is under strong selection because it is an important function for the organism, then you will see differences, and DNDS will pick up these differences. Okay. So these extreme cases where there are very large differences in the magnitude or the nature of selection acting on any two different sets of loci, those you will pick up, and you can pick up reliably with DN by DS. The issue is the large fraction of sites which are going to be in the middle, right, which are sort of neither extreme, neither is there strong purifying selection, neither is there strong positive selection, which is where most sites are going to fall. That is the part that becomes difficult to tease apart with DN by DS because of these different assumptions that then have to be looked at. What I'll do is on Slack, I'll post the paper that I'm referring to from Krajewski and Plotkin, and you can take a look at that.
Yeah. Is there something called moderate selection? Like, I have seen in some papers where people mention about moderate selection, purifying selection, strong purifying selection, then a positive selection, and kind of moderate selection. What do they mean by like moderate selection? Is it something between or?
Yes, so basically not very strong, right? So your values of DNDS, for example, may be higher than 1, but just about slightly higher than 1, right? Whereas a value of 10 would suggest that the number of non-synonymous substitutions that are fixed is 10 times higher than synonymous substitutions that are fixed. That would suggest very strong positive selection acting on these non-synonymous substitutions, right? Whereas a ratio of like 1.5, maybe relatively milder selection, perhaps.
Yeah, thank you.
Just a small thing.
Yeah.
Just by definition is going to look at sites under selection for protein coding loci.
Yes.
So it doesn't, it wouldn't really look at selection on regulatory non-coding sites, right?
No, this is only for coding sequences, yes.
No, thank you.
So for non-coding sequences, effectively people have developed other metrics that measure polymorphism levels, right? And ask how much polymorphism do we see at these sites in a population. And the general idea would be that using site frequency spectra, so thinking back to my lecture from last time, if you look at the allele frequencies in a population, you expect most alleles should be rare, right? Because they are going to be drifting around and will be lost or it doesn't matter. So you can look at site frequency spectra at these non-coding loci to then make inferences about potential selection or not. So basically look at allele frequencies in the population.
I see, but then I suppose it's not trivial to, think about what might be a positive change versus a negative change for regulatory regions, because
it's harder, yeah, unless you have specific models. So for example, in regulatory cases where in regulatory sequences, let's say, there's a transcription factor binding site, right? In the upstream region of a gene. Then and if there are empirical data showing, what this TF transcription factor what is that binding region and where it binds. And then you have a specific model that says, okay, an A here is going to be a good idea, T here is not going to be a good idea, and so on. So when you have specific models like that, then you can have a better handle on saying, okay, what is going to be beneficial or not. But if not, in the absence of that, so all of these, by the way, many of these very general measures that people have come up with, they were all developed, in an era where we simply had no idea what was going on in terms of function, even for coding sequences, right? So even DNDS is completely agnostic to the fact of which mutation is happening. For example, it's not distinguishing between two different non-synonymous mutations, right? It's just saying in general, synonymous and non-synonymous mutations should behave differently. And it's lumping all non-synonymous mutations in one category, which we know for certain it's not always going to be correct, right? And which non-synonymous mutation matters more than others is also going to change depending on the protein, depending on what else, and so on. So these are all very broad and very abstract measures to say, okay, statistically, can we tease apart, some signatures of selection without knowing anything about the function necessarily?
Sure, makes sense. Yeah, thanks.
Okay. All right, good. So here's an example. I'm going to just highlight a couple papers here that I found really like beautifully written papers, especially. So if you want to understand more and read more about this kind of work and these kind of analysis, I strongly suggest looking at these references. The couple papers I mentioned for this part of the talk, I just really love reading them. So, okay, so what do they do here? They looked at different Drosophila genomes. Okay, I think maybe they did some more sequencing of their own, but they're looking at two sets of populations. One is a particular lineage of Drosophila, I guess collected from Zambia, and so that's what it's called. And then one is this DGRP, which is a set of inbred lines that people have created as a resource, as a genetic resource for Drosophila. And people have a very large sort of sequencing database for these, right? And what they're asking is they're looking at fourfold degenerate sites, which means sites in the genome, encoding sequences, where you find synonymous mutations, where any of the 4 bases at that third position are going to lead to the same amino acid. Okay, so there's basically the maximum degeneracy possible at that site. And so they're focusing on those because there are many more of those in the genome. So you have a higher sample size. Okay, so you're comparing the fourfold degenerate sites, which should be synonymous sites, which should technically evolve under drift, and the short introns. And so those of you who are not biologists, introns are just pieces of DNA in a coding gene in eukaryotes, which are removed when the RNA is being transcribed, right? So after the RNA is transcribed. So imagine this is your entire gene. There are going to be little bits and pieces of that gene which are not actually going to code for protein at the end. Okay, they're there in the RNA and they're spliced out, they're removed. And then the rest of the pieces are called exons and they're joined together. And that is your mature RNA, which is then going to get translated. Okay. So introns, because they don't end up getting translated, are also expected to evolve under neutral evolution, right? Under drift. And so they're saying, well, maybe short introns, especially tiny ones, which get thrown out and presumably don't serve any real function for the most part, those may be a better way to estimate neutrality. And so they're comparing these short introns with the fourfold degenerate new codon sort of sites, so 4D sites. And the red populations here, red data are for Zambia and blue are for another line. And so these are inbred lines. And so it kind of makes sense already, hopefully, to all of you, that the polymorphism levels at these in these lines is lower, right? You expect to have less variation in inbred populations in general. So good, that is fine. And so then they ask, what is the relative level of polymorphism at these different sites, right? Now, what you notice is in both of these cases, the synonymous sites have actually lower levels of polymorphism compared to the introns, okay, slightly lower. Interestingly, if you now go and look for the sites where there are preferred codons that are being used, preferred meaning codons which are relatively commonly used in the entire genome. And the idea is that if that frequency of use is an indicator of some degree of positive selection, then you expect any selection acting on codons to be stronger at these sites, right? So they're focusing then on sites where preferred codons are being used at the moment in these populations. And they find that this difference becomes a little bit larger. And that happens in both sets of populations. And finally, and most interestingly, if they look at the unpreferred codon sites where there are already codons which are rarely used, they don't see a difference at all. Okay, so what this indicates is compared to introns, which should evolve neutrally as far as we can tell, at least the short ones, which because some introns can also encode other information, such as the splicing site and so on. But short introns are thought to not encode such details or useful stuff. Compared to those, synonymous sites are actually showing lower levels of polymorphism. And what's also interesting then is if you look at the ratio of the polymorphism in SI versus the 4D sites, so that's the y-axis here, you find that that ratio is higher when the codon usage for specific codons now increases. So what does that mean? For preferred codons, which are used more often in the genome, so towards the right of these plots, the polymorphism ratio, is higher. What does that mean? That the relative polymorphism at basically the difference between these two is stronger, right? And you see that again, this very strong positive correlation for both cases, suggesting that there is actually stronger selection for specific codons to be used at specific sites in the genome. And you see signatures of this in both of these. And so the way they've done this analysis is much more powerful than prior analyses. And based on this, they're kind of estimating that about 20% of all of these synonymous sites in Drosophila populations are actually evolving under fairly strong selection. Okay, Farhan, question?
Yes, Deepa. I don't understand how is that a polymorphism level for introns changes between preferred and unpreferred codons?
I think what they've done here actually is, you're right, you can't define codons for introns. What they've done here is they've taken each codon and matched it to, so it's a pairwise analysis for the nearest short intron site that they find, where I think they look at the same context. So like, for example, if your codon is AGG, your focal codon of interest, then they're asking for sites in the intron, which would be AGG. Of course, the triplet makes no sense in the intron, but they look for that. So that they're then accounting for differences in baseline mutation biases and so on and so forth. That makes sense?
Yeah, okay.
Thanks, good point. Okay. All right, so so this indicates that in Drosophila melanogaster, where people had earlier shown pretty weak levels of selection for specific codons, if you do analyses a different way and you find another set of loci which should be evolving under even more neutral selection, which should be evolving even more neutrally, you find that there is a difference between, you are able to now detect signatures of selection acting on synonymous sites as well. Here's another very nice paper and interesting work. Amir, question?
Yes, in the previous plot, so the absolute values for, say, preferred codons is lesser than for unpreferred codons. I couldn't understand why that would be the case.
Not lesser, no?
It is not
I see. So on the polymorphism, you're saying?
Yes, yes. If they're more selected, I don't know. Or I don't know how these values are calculated, so maybe.
Yeah. I'd have to think about that. Not sure. It might be that loci where they have unpreferred codons are generally having a lower mutation rate, because that's the case for even the SI, right? The value
is something like that, yeah.
So I imagine some such difference is what's causing it.
Okay.
Okay, so here's another really nice study, and this this brings me to a bigger sort of use of this codon selection, base selection. So many years ago, people proposed that, hey, if there's one way we can de-optimize viruses to create attenuated strains for vaccines, that would be awesome, because maybe if you make like 1,000 different synonymous changes to viral genomes, it would be hard for them to recover from that, right? So people have proposed this codon de-optimization as a strategy to make sort of very bad viruses, which cannot replicate well, but which can be used to elicit an immune response and therefore be used to generate vaccines. So there's been a lot of research on this. I'm not going to go into the details here, but I like this study. It's a recent piece of work because they try to tease apart now in different viral genes what happens when you de-optimize codons. And then they go on to do a lot more work, which I'm not going to discuss today. But I just wanted to share this as an example of real life scenarios where this kind of selection on codons could be useful in a practical sense, potentially. So what they've done here is they've taken this Phi-X, which is a bacterial phage. It infects bacteria. It's a virus infecting bacteria. And they've taken its genome, which has a bunch of these different genes here, right, shown in the letters on top here. And some of them are overlapping. I think I mentioned this briefly in response to some question on the first lecture. And then they ask how many of these and which of these genes have codons which are matching the codon usage of the host. So remember, viruses, they have to infect the host and they have to use the host cellular machinery. And so many times, though not always, we find that the codons used by viruses are a pretty good fit to the codons used by their hosts, which they infect. This is not always the case. So there are clearly other things going on, but we often find this the case, okay? So now what they've shown here is the relative usage of different codons. Wait, this is not making sense to me now. I think it's the codon. Yes, it's the, sorry. This is the codon adaptation index in the on the x-axis of this histogram. So in light gray, you're seeing all E. coli genes, and you're seeing that there's some distribution. So some genes have a better adaptation with respect to the tRNA pool that exists in that cell, and some don't. And then if you look at the highly expressed genes in E. coli, which are the ones shown in black here, they tend to have a higher codon adaptation index value. This is on the x-axis, which is an indicator of how likely is this gene going to be expressed rapidly translated rapidly, because there should not be problems with waiting for rare tRNAs to come and do the translation. Okay, so what they do then is they take the 5X wild type genes, and in a bunch of these genes, they create variants, which are synonymous variants, so similar to what I showed you yesterday last time for some of our work, but then they de-optimize them a lot. So that now, for example, here's a gene C, which had this particular CAI value, and they've created different versions of that gene, which take it to different optimized or de-optimized values. Okay, is that part clear to people? So they take the same gene J and they make one very optimized version, which should be better matched to E. coli genes, and one version which is super de-optimized. So it's at the tail end of how bad E. coli genes can get. Okay, and then they ask what happens to fitness of these phages, which they have either optimized or de-optimized. And the way they measure their fitness, the phage fitness, is they allow them to infect E. coli, and they count how much effectively do they have, right? And so that's the plot I'm showing you here. So the horizontal line is the wild type. And so I think this gene C is representing what happens with the wild type if you don't make too many changes here. Now, if you look at all these different genes which they've optimized or de-optimized, one thing you notice is they all are kind of bad compared to the wild type, right? So any kind of change in codon usage is making all of them worse compared to in terms of infecting E. coli compared to the wild type phage. But some of these versions are better than others, right? So each dot here of the same color is showing you a different codon version of the same gene, right? And so this one happens to be optimized, and these are all de-optimized and similar. So overall, they do see a pattern. I'm not showing you all of the data in this paper. What they do see is a pattern where if you have versions where they have de-optimized these genes, they are worse off. The versions where they have optimized the genes are better, but they're all still bad compared to the wild type. And they find that the fitness is nicely correlated with how many rare codons they have in the population, in the gene, as well as with the mRNA structure and the folding and so on. Okay. Question?
Deepa, when you're optimizing in the wild type, what are you doing? Like, in terms of de-optimization, you're what are you doing in terms of optimization?
You're increasing the codon usage such that it matches that of E. coli codon usage, the highly expressed E. coli genes, right? So basically optimization is, if you look at this plot here, optimization is all of these are on the right side. So they have a higher codon adaptation index. They look a lot more like the highly expressed genes in E. coli. And the de-optimized ones are all towards the left, right? So that they are all having lower CAI values.
Okay. Yeah.
So the prediction is that the de-optimization should reduce the ability of the host cell to translate these mRNAs, and then the other way around for optimized versions. Okay. And so, like I said, I like this paper a lot because they do a really great job of discussing the history and the background and so on really well. So I would recommend that if you want to know more about this, this kind of work. And then in this particular study, they also go on to ask what happens if we now combine different pieces of these de-optimized or optimized codon versions. So A gene is optimized, but D is super de-optimized. So basically trying to understand epistasis as well across these different genes. And there's interesting data there as well. Okay, so I want to now move to talking about a different set of assumptions. So this is all to say that we've sort of made assumptions about synonymous mutations and selection at synonymous sites for decades, and that has been the baseline on which we've built a lot of the kind of methods that are used a lot in population genetics and evolution. And we need to look at those and rethink those assumptions a little bit more, and hopefully develop new methods. And that's something many of you maybe can contribute to figure out ways to detect selection, which are moderate levels of selection. So like I said, for extreme levels of selection, I think these metrics will still work fine, because we do see evidence that synonymous sites do on average experience weaker selection compared to non-synonymous sites. But that's not always the case, and that's the issue. Okay, so I want to move now to another set of assumptions, talking about understanding what happens under selection and what happens under drift. So you've heard from many of the earlier talks about evolution under drift. How do we empirically address this, right? So selection seems a little bit easier. I think most of us have an intuitive understanding of how would we detect what happens under selection, right? So we take populations, we might evolve them under selection, we put new selective pressures on them and ask, okay, now how do you adapt, right? And that's a sort of straightforward way in some sense. Humans have been doing this for centuries. As we domesticated plants and animals to get varieties and genotypes, and which would be beneficial for our uses. So what about understanding? But to understand what happens under selection, you need a comparison very often of what happens in the absence of selection, right? To be able to understand what is the baseline in some sense, and then compared to that, you understand selection much better. So people use what are called mutation accumulation experiments a lot. Here's an example of what happens. You're basically passaging your population through a series of bottlenecks, ideally having a single individual at each bottleneck, so that every set of transfers or every few generations, you're basically making sure that only one genotype is able to pass on to the next generation. And that means that your population is going to evolve under very, very strong, very weak selection, and primarily under drift, right? So your effective population size is 1, and that's what you're trying to ensure. So here's an example with E. coli plate or bacterial plates. You take your ancestral plate, you randomly choose one colony which you're going to transfer to the next plate, and so on, right? And because you're choosing randomly, not based on whether this colony is big or small or any such thing, you're going to end up accumulating random mutations. Now, what mutations are going to be missing in this kind of scenario? Of course, you will miss the lethal ones, because then if a colony, let's imagine the cell was transferred at this spot, but there's no colony here because the ancestor, let's say, had a very deleterious mutation, it couldn't grow, then you're going to miss those kinds of mutations because they will never accumulate in your experiment. So that's a problem. Outside of that, the idea is that this allows for a sampling of mutations to be pretty random. Okay, so this strategy is used for many different sets of questions in evolutionary biology, and I'll discuss a few. Okay, so Muller's ratchet is this concept that is this phenomenon that's predicted to occur when you evolve populations under drift like this, right? So if it is true that most mutations are deleterious, you expect that as populations accumulate mutations, right, most of the mutations that are accumulated are going to be deleterious. And so you expect that over time, under such a regime, fitness should drop. Okay, so this is one of these early studies showing that indeed does happen. This is with viruses. This is what they did. They take, at time 0, they take a bunch of virus, they allow it to infect a plant. And then wherever you see these holes, it indicates that the virus has infected the plant tissue at that point. So you take one of those points, so it's like a bacterial colony, and then you allow it to transmit and so on, right? So they did this experiment for a bunch of different transfers. And indeed, what they find is if they plot the Malthusian parameter, that is the exponential growth rate of these phage colonies, not colonies, but I guess, what are they called? Clearings produced by the phage as it eats up the tissue, plant tissue. You find that the rate at which that growth is happening of the phage particles is dropping over time. And so each of these is an independent experiment that they did, right? And so this sort of indicates that this ratchet, which is this irreversible process where once you're accumulating mutations, in the absence of sex and recombination, you cannot purge these mutations, right? You're stuck with these mutations. So basically, over time, lineages will continue to accumulate one deleterious mutation after another. Once in a while, you will get some beneficial mutations, of course, but if the general prediction is that most mutations are deleterious, your fitness should decline, right? Similarly, people have also done experiments with C. elegans, so eukaryotic species, where they've taken a homophyletic ancestor of C. elegans, these worms, and they've done interesting things where they've asked what happens if the bottleneck size is different, right? And so a simple prediction is, which I hope most of you find intuitive, that if your bottleneck size is larger, then the rate at which that ratchet is going to click should be weaker. It should be slower, right? Because you're basically not allowing the bottlenecks to be so severe. So they tested this. They had bottlenecks of either 1 individual or 10 or 100 individuals at different transfers. And then they compared what happens to the productivity. So this is just how many offspring are being produced by each worm, measured at the end of this experiment. So after mutation accumulation generations, 100, 200, 300, 400, you find that maybe there's a slight trend in all these larger population, bottleneck size populations. But in the n equals 1, where there's a very strong bottleneck imposed, there is indeed a rapid reduction in fitness, not a very strong reduction in fitness in the other populations. And then they have a control line where they don't do the bottlenecks, they just maintain the line at some high population size. Okay, there is evidence showing that indeed if populations evolve under drift, and these are just a few examples, there are many, many more examples. If populations evolve under drift, they will typically lose fitness over time. Pratyush?
Yeah, Deepa, I just had a question. So is this experiment with C. elegans done asexually? Like this is like selfing kind of thing. So there's no purging of deleterious mutations because of recombination.
Yeah.
Okay. Yeah, thanks.
Any other questions? Okay, good. So people have used mutation accumulation experiments to understand what happens under drift. And in some cases, there are some very nice experiments people have used what happens with bottlenecks if you have populations evolving under drift, and then you ask what happens in new habitats when populations face selection. How does that compare with what happens under drift, right? And so that comparison gives you a very nice measure of what is this power of selection to cause phenotypic change and so on, right? Because then drift becomes your baseline comparison. So now the only problem is there is some selection bias that can sneak into these mutation accumulation experiments, especially when it comes to microbial populations. So let me give you an example of what I mean. Let's see, here's a plate. Like, imagine there's an agar plate on which we have bacterial colonies. And like I showed you before, we're choosing one colony at random to transfer for the next generation, for the next plate. What happens is at the center somewhere of this colony is the very original cell, right, that started that colony. Now, if that is growing, radiating outwards as it grows, as you've seen in David's talks for the last 2 days, you can form sectors like this, where a beneficial mutation, if it arises at this point, is going to spread in the population, and it's going to occupy a bigger chunk of this pie than what it would occupy if it was a neutral mutation, right? So deleterious mutations are going to suffer the exact opposite fate during this growth of the colony, where they will be outcompeted by either other beneficial mutations that occur next to them or just the wild type lineage with no mutation, right? And so the chance that a deleterious mutation is lost is going to be higher. And a beneficial mutation, if it occurs, is the chance that it spreads in that colony is going to be higher as well, right? So what's going to happen is now if I'm going to pick most of this colony, okay, I take up the entire colony and I spread it on the next agar plate for my next set of growth, I'm going to get a little bit more colonies that are carrying the beneficial mutations compared to the gray ones or the deleterious ones, right? Not compared to the gray, but compared to the deleterious ones, right? So how do we account for this? And is this truly, how important is this effect, right? So remember that what we've assumed when doing mutation accumulation experiments with microbes as well is we've assumed very, very low amounts of selection or very weak selection. And this kind of problem is going to occur because of growth that happens in this phase, which is necessary because at the single cell level for microbes, you can observe them, right? So you can't transfer them, which means you have to wait until it becomes a visible colony, and that takes some 15 to 30 generations of growth usually. So let me give you a sort of cartoon picture understanding of what this means and why we need to correct for it. So here is a potential selective effect of mutations. Just 0 means no effect. So it's a neutral mutations in gray. Plus means positive, and minus means deleterious mutations, right? So beneficial mutations here are in green and deleterious are in purple. And you might draw a DFE that kind of looks like this. There are some beneficial mutations and a bunch of deleterious mutations, right? So this observed DFE could be a little bit more symmetrical, as I've shown here in this cartoon. But now, if we say that there is selection bias that is occurring, effectively what is truly the DFE is that there were many more deleterious mutations here which were lost during the colony growth, and there were fewer beneficial mutations, relatively speaking, as a proportion of the total, and we have overestimated that amount, right? So if I want to correct the DFE now, what I need to do is somehow figure out a way of increasing the height of these bins in the histogram and reducing the height of these bins in the histogram. And that's kind of what we try to do in this work that I'm going to describe to you now. So this is work I will qualify by saying this is, the maths here is all done by my collaborator, Lindy, who's also been an instructor in this course twice now. And I was very fortunate to get started on this collaboration, the roots of which are in another project, which we started in the last PopGen school. Okay, good. So let me walk you through how we are modeling this, right? So what Lindy does is she says, okay, let's assume exponential population growth during the growth of a colony, right? So your population size at time t is going to be the population size at time 0, which is what your starting population size was, and that's going to be 1 when we're doing an imposing and bottleneck of 1 for the MA accumulation experiments. And it's exponential growth determined by the rate of division. So RT is going to give you the rate of division. And so if it's a simple doubling, like we assume is happening during exponential growth in bacterial or other microbial populations, then you can write this as N0 to the T, right? So cells are dividing and doubling, and that exponential growth is captured here. It's a simple formula. Now, the rate at which new mutations occur, let's imagine, is mu. And that mu is going to get multiplied by our population growth to say at how often am I going to find new mutations during the growth of the colony. The growth rate of the mutant lineage is determined. So we're basically saying if mutations are deleterious, the growth rate is going to reduce, right, by a value which is proportional to the selection coefficient of that mutation. If it's a beneficial mutation, that growth rate is going to proportionally increase by a similar amount. All right. Now, what is the size of the mutant lineage in time tau, where time tau is the number of generations or number of doublings that could have happened during the growth of the colony. So this is going to get, this modification to the growth rate is going to go and get plugged in here in our exponential population growth equation. So that's where it gets plugged in. And tau minus t is the time that has, if t is the time when the mutation occurred, tau is the time which when we're finally doing the next bottleneck, right? So that's the amount of time that that lineage has, that mutation has to expand or contract in the population. Okay, so this is what happens in proportion to the time it has. Okay, so then what we can do is we can calculate the relative bias imposed by selection. So this is the bias as a function of the selection coefficient of that particular new mutation. How do we calculate this? We basically say how often is this mutation going to occur at time t, and what is the size of the lineage of this mutation at time tau, given that tau minus t is the time it has available to expand. We integrate that over all of the bottlenecks that have occurred, right? And the same thing we're doing is in the denominator here. So this is when we're saying S is not equal to 0. So it is either a beneficial or a deleterious mutation. And in the new denominator, we're saying, let's compare that to the situation where it's a neutral mutation, where S is 0, right? Because this is the scenario where there is no difference in the fate of this mutant compared to the wild type, if it's a completely neutral mutation, right? So basically normalizing how much is the population of this mutant going to change or going to increase or decrease relative to the effect of a random mutation, which is neutral. And so this, we plug in these equations here, and ultimately this simplifies a little bit more. If we assume that the mutation rate is much slower than the growth rate, and this is a fair assumption in this case, because the growth rate is, many doublings in per unit time, which is between transfers, the mutation rate is much lower than that, right? So the chance at any one site is going to change is on the order of 10 to the minus 8 or so. So it's a much smaller value. And so this simplifies to this quantity. And
one question.
Again, plugging, just one second, let me finish this. And if you plug in this this way of writing the exponential growth in terms of doubling times, we get a simpler equation where the relative bias due to selection for or against a particular selection coefficient is going to be a function of the selection coefficient, right? Times the time that it has had to, that lineage has been hanging out in the population, right? So it's proportional to that time. As you can imagine, as the time increases, if you allow for more generations of growth during colony expansion, it has a higher chance to accumulate in the population if it's a beneficial mutation. The higher the value of S, that means the stronger the selective effect of that mutation, the bigger the chance that it is going to spread in the population. And the denominator, we have those along with, this is what happens when there's a neutral mutation that's going to come up. Okay, question?
Yeah, I was just wondering that, you've chosen the mutation rates to be same, irrespective of what kind of mutation it is. So for the neutral ones, beneficial and deleterious, but in general, they are different, no? For example, beneficial mutation rates and the deleterious ones, they can be very different. So isn't that going to affect things?
Yeah, so in this particular equation, we're agnostic to what is the fitness effect. When we do the simulations to test this, then of course we add in different mutation rates or we can add in different mutation rates for deleterious or beneficial. This is saying for any one given mutation, given that it occurs, what is the outcome?
Yeah, okay.
Any other questions for this so far? Okay. Amir?
So the limits that you have put here, that's just for like one transfer, is it?
Yes, because the colony growth is happening in between that, right?
Okay, so it's just one transfer and the selection bias for one transfer. Okay.
Yeah, but no, so it happens in one transfer, but then if that, so in proportion to the lineage expansion during that one transfer, this is the degree to which, this is the degree to which we're going to have an overabundance of that particular mutant in the colony that goes to the next cycle, right? And so now if that colony makes it to the next cycle, then this process has to be reiterated, right? And so on.
Yeah, also this is for just like one type of mutant, right? Because your S is constant.
Yes, yeah. So you would basically need to do this for every mutation, every possible value of S if you wanted to actually understand what it's going to do. So I'll come to this next. Okay.
Yes,
okay. So how do we now correct for selection bias during mutation accumulation, right? So if we believe that this process is happening for all kinds of potential values of S, depending on how many growth cycles have happened, how many transfers we've done, and so on. So the weight of the fitness bin I in DFE, in the DFE histogram is giving us a selection bias. So remember in this cartoon earlier, I'd said, if your raw DFE is shown by this dotted line here, right, which is a lot more symmetrical on both sides, what we want to do now is we know that what's going to happen is that the deleterious mutations are going to be underrepresented in this DFE that we observe because they will be lost in the population. And the beneficial mutations are going to be more likely to be observed in our raw DFE because they are more likely to hang around and make it to our final population when we get single mutants. Okay, so a corrected DFE then has to somehow suppress these bins in the histogram and somehow elevate these bins in the histogram. And so what we're doing here is we're calculating by how much should we weight these different bins, right? And that weighting should depend on the relative selection bias that's going to come in, given the value of S, right? So for any given bin here, if these are all denoted by I, so the average fitness of any one bin is Wi, and the average selection coefficient for that bin is Si. And so what we're going to do is basically weight it simply in proportion to how much of a selection bias we expect given the value of S. So if Si is large, we would give a larger weightage, right? So this bin should then drop by a lot. If S value is relatively small, we're going to do a weighting which is proportional to that, to the amount of selection we expect there, okay? And so based on this then, what we do is we correct the frequency of each bin in our final DFE histogram, basically by normalizing so that all frequencies add up to 1 in the end. Now, one thing I want to highlight is that this is all done only assuming there is a single mutation accumulating in any given lineage, okay? We don't yet have a correction for what happens when there are many mutations that a lineage may accumulate. We're doing this only right now for a single mutation. And the multi-mutation problem is that epistasis will start playing a role, and it's much harder to account for that. So at the moment, this is a simpler model. We don't account for it. So what we're assuming is we're stopping the experiment whenever we get a single mutation. Okay. Any questions so far? I'll show you now examples of what happens when we apply this. Okay. So here are simulations then that Lindy did with the basic calculation of how much selection bias do we anticipate, should we see, depending on the selective effect. So the selection coefficient on the x-axis, 0 means it's neutral, and the relative bias that we calculate based on that equation, given the S value. And what she's done is she's done simulations to ask what happens if there are 5 generations of growth, 10 generations of growth, 20, 30 generations of growth between each bottleneck, right? And so what you can see is in general, as your selective effect increases, so these are strongly beneficial mutations, they are more likely to be they are more likely to generate bias, right, in your outcomes in the end. So they're likely to be proportionally overrepresented in the population. And that degree of overrepresentation changes or is higher when you have more generations of growth. So the red lines here with 30 generations of growth are ramping up much faster compared to the green line, which is 10 generations of growth, right? So that kind of makes sense intuitively, that you'd expect as there are many more generations for a beneficial mutation to expand, the effect is going to be stronger. And the more beneficial the mutation, the stronger the effect is going to be. Now, if you look at the relative, and another way to look at this is just on a semi-log plot. So the right side plot here is just a semi-log version of this. So you can better see what happens at the deleterious end. And it's exactly the opposite of this, right? So there's a stronger effect. You're more likely to lose more deleterious mutations, and that effect is going to be stronger when you have many more generations to lose them effectively, right? Now, at the bottom here on the left, you're seeing what happens as a result of the selection bias to simulated DFEs. So let's imagine we start with the true DFE, which is shown in black here by the black line. And let's say for the purposes of the simulation, this DFE is more skewed towards deleterious mutations. So it's on the left of the 0 line. As you increase the number of generations between bottlenecks until you acquire one mutation, you see that you get a stronger and stronger skew towards more beneficial mutations. So the DFE is shifting towards the right. And if you now normalize this so that all the frequency distributions have a similar probability density, you start seeing patterns that look more like what we're used to seeing for DFEs, where all these DFEs have the same area under the curve, and basically they're all shifting towards the right? And that right shift is stronger as you have more generations and more time for biases to evolve. Okay, sorry, biases to propagate in the population during the colony growth. So here is another way of looking at this. Again, these are simulations. So if you have a true DFE, which is shown by this black line here, and an instance of what you would observe shown in purple here, if you allowed the selection bias to perpetuate, then you end up with this purple histograms, and the purple histogram is much sort of towards right shifted compared to the black line as you have more and more generations of growth. And if you now apply the correction that I just showed you the equations for to this purple histogram, you start recovering much better the blue histogram, which is much better reflecting the actual true DFE that underlied this simulation. Okay, so each result here is an average of many different simulations that she did for each set of parameter values. Okay, and so here is what happens as a function of growth between transfers, so the number of generations on the x-axis. How does the fraction of different categories of mutations change? So here's the uncorrected, which is the stars shown here. So as the growth between transfers increases, you find that the total fraction of the DFE increases between generations. The mean selective effect goes up. But if you now do a correction, which is the circles, you find that the corrected DFE shows that the deleterious fraction or the beneficial fraction in this case here does not change across different growth parameters, right? Whereas as the growth between transfers increases, as your mutation accumulation proceeds, you find that your average selective effect of mutations goes higher and higher as your growth transfers increases, right? Whereas if the actual distribution, like if there is such a thing as the real underlying DFE, it should not change as the number of growth cycles increases in your experiment, right? And so the correction works, and because it basically stays constant, these values, and the correction is useful then, and it's more accurate. It matches the true DFE much better, okay, which is shown in the dotted lines here. Questions?
Deepa?
Yeah.
Between each bottleneck, our generation time is not constant, right? Let's say you picked up a bigger colony in the first instance, and in the second instance, you picked up a smaller colony, right? So that would change the DFE, like it would shift more towards the deleterious one in the second case, where in the first case, it's shifting to the beneficial.
Yeah, so we're not accounting for that. We're assuming there are not many differences between the number of doublings in each transfer.
Okay.
Yeah. And also I should mention, so what's happening here is in these simulations, she's not assuming that there are multiple transfers happening in the bottlenecks. Okay, so it's not actually mimicking a true experiment. What she's doing is she's just saying, let me assume X number of transfers and then iterating like hundreds and thousands of times so that in some colonies, there will not be any mutations. In some colonies, there will be mutations. So the way the simulations are done, they're not exactly mimicking an actual experiment, right? Where you take that linears and allow it to go forward and so on. Because here, like I said, we're interested only in the scenario where there's a single mutation that could be accumulated in that one growth cycle.
Okay, yeah.
Yeah. I should also say that, yeah, there's a little bit more nuance there, but maybe I'll wait and see if there are questions, but there's a lot more that's worth reading in this paper. If you're interested in this, I would urge you to read the paper more. But this is not mimicking exactly what happens in actual experiments. All right,
sure.
So here's a case where we applied the correction for selection bias. So I think I showed you some data for DFEs that we had calculated or estimated based on single mutations that we accumulated in MA lines in E. coli. This is work done by Mridula, who's also attending this school. And we got raw DFEs shown in purple here, which were surprisingly skewed towards beneficial mutations. And so we applied the correction shown here in cyan, and then we get this DFE. And whereas earlier we had about 40% mutations being beneficial, so this is this blue star in our earlier DFE. if we apply the correction, we now get a number which is much more in line with expectations from theory and from other data, where we have lower, less than 5% beneficial mutations or so. And similarly, the deleterious fraction of mutations increases from only about 30% to more like 60%. And this is in a rich media like LB, where we do expect the proportion of beneficial mutations available to E. coli to be much smaller. And so similarly, the average selection coefficient for all these mutations that are accumulated also reduces with the correction, that overall these really big selective effects are sort of now squished, and you can see that the DFE is squished much more between a smaller range of values because of these corrections that we're applying. Okay, so the upshot of all of this is that we've thought of mutation accumulation studies as a great way to weaken selection and minimize selection, but there can still be substantial selection that's acting. I should also say that it's not that means that MA lines are useless and do not reduce selection. They do. So all of this is saying that, yes, there is still some degree of selection, but it is still definitely weaker than what we get in traditional selection experiments, where we impose very strong selection on whatever trait of interest or whatever metric that we're evolving the population for, right? So MA lines are still a great way to reduce selection. It's just that we have assumed that selection is nearly zero, but that's not correct. And I should also point out that we are not the first people to say, well, there is no selection. There is selection during MA lines. People have known this, as far as I know. A paper by Mike Lynch and Kibota were the first people to correct or offer a correction for this kind of bias, but they focused only on deleterious mutations because they didn't expect the beneficial fraction to be large enough to be important. And so in a paper in the 90s, they offered a correction for the deleterious rate. Other people have done other forms of corrections, again, focusing largely on the deleterious rate. And the only interesting example is from Supreet's lab, which also came out last year, where they also did in parallel very, very similar simulations to what we have done. And they also show that during MA experiments, you can have substantial selection. And what I really liked about this paper is the nice set of experiments they did to test some simple predictions that come out of this model, right? So what you expect then is if your colony size is large or small or medium, you expect that selection bias should then play a bigger or a smaller role because there are more or fewer generations of growth. So I guess this addresses a little bit of your question, perhaps. So they're saying is as the number of transfers increases during the MA populations, small colonies, if you're only selecting small colonies, they are less likely to show drops in fitness because there are fewer generations in which the selection bias can take hold, right? And you see bigger drops in fitness as your colony size increases. And what they also did is asked, again, the simple prediction is that if you're collecting your cells for bottlenecking for the next transfer from the outermost part of the colony, you might be more likely to pick up some beneficial mutations. And so they did that. They took large colonies where they looked at this, they only took the center of the colony to propagate, or they took the edge of the colony to propagate. And if you propagate the edge of the colony, you find that the growth rate is higher, right? That's where you expect to see beneficial mutations in larger abundance. And so do look up this work as well. And if you have questions, get Supreet, who's around here, about this work as well. Okay, and then I want to end this session today by saying or pointing out that even if we correct for selection bias, we still often find a much higher fraction of beneficial mutations than expected from theory and observed in some sets of populations. So I'll just give two examples here. This is data from 2004 paper by Joseph and Hall, where they looked at MA lines of yeast and they measured fitness in these MA lines propagated for some hundreds of generations. And you'll see their DFE is showing a large number of mutations or large number of lineages have beneficial fitness effects, right? And in Arabidopsis, similarly, this is a more recent paper where people have done MA lines in Arabidopsis, where the problem of selection bias on colonies does not exist or should be very, very small, because there you can actually have single individuals during the bottlenecking. Here, if you look at the probability density of fitness effects of mutations, and they've normalized this by the founder fitness because they also have some variation in the founder fitness itself. So just a normalized x-axis here. It's still true that the right side is beneficial. You again find that lines that are tested at home, which is their sort of native habitat, versus away, which is a different habitat, you find a substantial fraction of beneficial lineages here as well. And on average here, they've estimated that their lineages have about 5 mutations accumulated. In this case, it's not known how many mutations are accumulated, but there are many of them, right? And so one of the issues with these kinds of populations or these kinds of data is that we don't know what is the impact of each mutation separately. So most MA lineages or most lines where people have measured fitness effects after MA have been done with multiple sets of mutations in each population, many times, most times unknown number of mutations. And so data I showed you earlier for DFEs are amongst the very few cases where we have single known mutations anywhere in the genome, and we are measuring the DFE based on that. People have such data for single genes, and there as well, in many cases, you do find a not negligible proportion of mutations are beneficial. So we still are left with this problem of, why do we see many more beneficial? We seem to see many more beneficial mutations or not so many deleterious mutations compared to what theory has assumed or has predicted. And so something is not quite matching up, and we need to think about this more. I don't have any answers for this really. I'll also point out that in this yeast paper, there's a follow-up paper from a few years later where they asked, okay, maybe we saw this because we only measured one fitness metric. And so for these same lineages, they measured a bunch of other different fitness metrics, and they all show the same pattern. They also asked, okay, maybe if we evolve them longer, then we'll start seeing declines in fitness. That didn't happen either. So they found that, the fraction of lineages which showed a relative increase in fitness was also not negligible. Similarly, in a bunch of other papers with yeast, with Arabidopsis, with I think other single-celled microbes as well, people have found that when you take MA lines and you evolve them for a long time, there is quite a number of them that show either no change in fitness on average over time, or they show an increase in fitness. That's not expected unless the fraction of beneficial mutations is actually pretty substantial and not a tiny, tiny fraction, right? So we do have this mystery still. We don't fully get it and understand it. It's clear that some of the issues may lie with experiments or with empirical. Like I said, we were making assumptions about how weak selection is, which turn out to not be negligible or turn out to be important. But there are probably also problems with how we have made some of these estimations of what the DFE should look like without having sufficient understanding of how many mutations went into these estimates. I think there are some questions. Please go ahead.
Deepa, so slightly unrelated, but in the Arabidopsis data, we see that the away DFE is shifted more towards the deleterious mutation. So I'm just slightly surprised, because in general, don't we expect that when an organism is in its native environment, then it is reasonably well adapted, and therefore it would have already sampled many beneficial mutations. So it's the new beneficial mutations available should be less, and therefore we should have the shift of the DFE towards deleterious, whereas in a new environment we should see more beneficial. So this seems to be the opposite of that. And do you have any idea why this could be the case?
No. So the authors don't have any idea either, really, for this particular paper. And so, yeah, we don't know. Of course, this home and away is defined in a peculiar way for these Arabidopsis accessions, like basically where they were collected from or where they were sampled from originally. And it's plausible that something has changed in the environments now, so it's also a very rough definition of what is home and what is away, right? And what is actually, I guess, optimal for growth for a particular organism. And so I think maybe it's because of the definitions of home and away and our sort of intuition about it, which is perhaps not quite accurate. And the details really matter in this case, potentially. So that's one possibility, but we don't know the answer really. Yeah. Kavita, you had a question?
Yeah. So you mentioned that, there are more beneficial mutations than expected on the basis of theory. So I was just thinking what kind of theory you had in mind. So the typical papers that I've seen in experiments assume that this beneficial mutation, the distribution is typically exponential is what people, assume. But this theory has been generalized by Alan Orr and others, like including our work. But so like when you say that, it does not match the theoretical expectations. So I just was wondering what kind of theory are you referring to?
Yeah, so I guess generally, so if you look at the review papers by Kitely and others, right, or even, well, I guess fits to the data. So not necessarily theory as in here is what we expect to see, because it's not, theory can't tell you how many you can find, right? Ultimately, it depends on the parameters that you add. But
the theory can tell you. So when you're saying fit of the data, but fit of the data to some theoretical function, so right? So you will try some fits to some function, and the job of the theory is to understand, rationalize those functions. For example, so one very popular one, and Alan Orr had started this in the 2000s, was that he gets this answer to be just exponential, and I think that was very, very popular. It became. But then later on, it was realized from some other theory that it should actually be a much slowly decaying function as well, much slower than
the beneficial side, right? Yeah. For the beneficial side of.
For the beneficial side, yeah. So for that only, I'm aware of some theory. So I'm not surprised that that people find more than what they expect if the expectation is exponential. So is that what you had in mind, or something else? So that's what I just wanted to clarify.
Yeah, so some of these experiments, yes, the exponential was used as the baseline. So for the yeast papers in the past, that's what they've used to say if there's a simple, I guess, based on some parameters that they observe here. If I fit a simple exponential, what would I see? And they don't see that. But when I say that expectations based on theory, I think I meant much more on the lines of when people have seen data for large numbers of mutations accumulated and not sort of knowing exactly how many mutations were accumulated, or you see from selection experiments, you draw DFEs and you fit the data to that, fit models to it and say what that should look like. That's, I guess, the kind of expectation that I'm saying, not necessarily for a data set like this, but saying that distribution doesn't match the simpler, I guess, predictions from prior work. I guess I should reword that. I don't know, maybe you can suggest how I should reword that.
Okay, just check out these papers. Thanks.
More questions?
I had one sort of thought seeing this. So coming back to what Susanna has been saying for a long time, that we have a lot of robustness and things like that. I was just wondering whether it's not that easy to find deleterious mutations that reduce fitness, because you have some kind of robustness to all these deleterious mutations, and you don't see their effect as much. And so that's why.
But then you shouldn't expect to see beneficial mutations either, right? Robustness would not mean necessarily that only deleterious effects are dampened. It should mean that any effect should be dampened. But
she was showing that under some kind of selection, even with neutrality, you sort of see a quick switch, some kind of transitions where you have, even with small changes, you get large beneficial effects just by very small switching. So whereas the other case, it might not be very true, because these large networks, they're sort of robust to deleterious changes.
I know, but my point is that the robustness to deleterious changes, I don't see how it would be one way, right? Like practically speaking, at the level of a network or a cell sort of evolving to say, okay, it's not going to be like, I don't want only deleterious changes to be buffered against. It should be, any change should be buffered against. That's what's going to end up happening.
Yes, but that could depend on how the gene network is placed at, right? If it's closer to something that could be beneficial, then that asymmetry would come about, and that's something that you might see in these kind of plots.
Sure, but I don't see how that could still explain what we see with MA lines, right? Where it's not that they are necessarily close to optima or not.
Yeah. I just like thinking whether how this could be.
I should think more about this. Maybe to some extent that's plausible. But what we do see, of course, is still typically the tail end of the towards the deleterious side is much, much bigger. And we still observe by and large that there are many more deleterious mutations than beneficial, usually. So, yeah, I can think about that more. Any other, somebody else had questions? Yeah, Kavita?
I know I asked already.
Please go ahead.
I asked already.
Okay, all right. Okay. Yeah, and please, I'm happy to get feedback about this as well. Maybe Kavita, I'll talk to you more again about this, but
it's nice. I like it. I mean, it's very nice that you showed in that, like, the previous data that you showed, and how you said that like 0.4 or something was selection coefficient. When you take this more sophisticated analysis, it drops more to the sort of more, that is sort of, comforting in some ways, right? So reassuring that, this is sort of something very, very different from what people have learned over the years. So it's nice to see that everything is still in place. So very nice. And congratulations.
But we do, but there's still a puzzling number of beneficial mutations. So puzzling, I guess, see, from my perspective, it's not really puzzling because we see it, right? So if we see it, then we see it. But I guess the puzzling part is really that, so like I said, we're not the first to even find a large number of beneficial mutations in our observations, right? People have found them since the 1990s. So older Arabidopsis work, which I'm not showing here simply because they didn't have nice figures that I could easily sort of explain and show. But work from Ruth Shaw's group and others around that time have also found a larger fraction of beneficial mutations than reported in other papers or discussed in sort of these review papers at that point. And so people have been finding, a substantial fraction of beneficial mutations, which has been surprising, and there has been no real explanation for it. A large number of ideas have been floated, but not really. And this particular plot, I don't remember if this plot has corrected for selection bias to some extent. I guess they did correct for deleterious bias based on the Kubota and Lynch sort of correction, but not for beneficial mutations. I should also mention that others have, a couple of papers in the past have used corrections based on a modification of a completely different model from Sally Otto and Maria Uribe, where they looked at somatic lineages and how they are expanding during somatic growth, and asking how mutation biases or selection biases will play out during that. And so they said, well, this is kind of similar to what happens in yeast colonies as they're going through MA. And so they modified their equations used for somatic lineages and how they're expanding with selection bias to correct for their estimates. And they also still got sort of pretty high estimates. So I do think there is still more beneficial mutations than generally what I see used in population genetic work. And we don't fully understand where it's coming from. Is it that there's a problem in the experiments that needs to be corrected, or is there a problem in somehow our expectation that most mutations are deleterious pretty much generally? And I suspect it's some mix of both, but we don't fully understand it.
Yeah, just can I mention just a couple of other works I'm reminded of? I think one is from Claudia Bunk's group, I think, Fall et al., FOLL et al. Okay. And I think one is Aryan's group with Joachim and some students. So in those two works also, the distribution is, very, there are large effect mutations, very large effect beneficial mutations, and quite a few of them. I wonder if you're aware of those two papers. I haven't kept track of literature after that. So if you like, I can give you the references.
I'll find them.
So those two are, I know. So, I think
one of
evidence are finding more and more these days because maybe better sequencing, better resolution, and so on.
Yeah.
So perhaps there is more.
Yeah. So that you're right. I mean, so there are cases, of course, of individual mutations being super high effect, and that people have reported. And I think that's not super unexpected. People don't, are not surprised by that. It's these moderate level selection coefficients but really high frequencies of those I think people are much more surprised by, or I guess are unexpected. I mean, we had another piece of work where we were showing these DFEs, and which is still in review now, and we got major pushback from reviewers for that work, because they're like, well, how do you see such high fractions of beneficial mutations? And so I think some part of that is this reduction. But when we apply this correction, not everything reduces to this super low level of 2 to 5% beneficial mutations. We still have situations where we have much higher fractions, and in environments which are not sort of terrible environments, so that you might expect a high fraction of beneficial mutations. So I should look at this work. I don't remember seeing Claudia or Joachim's work where they've described the DF itself.
I just have one more small question, which is, so I thought that, these things people have been analyzing for like ages meaning like 20, 30 years or whatever. But there must be nice software developed where all these corrections are already taken care of, because, some people do this a lot. So, I mean, isn't there some sort of standard software I can feed the data and it takes care of all these things? There's no sophisticated softwares like this already?
Not as far as I know. No.
Because, it's very difficult, right? Because if the numbers drop by such large percent, okay. Maybe.
Yeah, no, nothing exists. I mean, even the correction, has for all categories of selective coefficients, we didn't have one before this. The only correction people used to apply is to sort of up the number of deleterious mutations that you expect, because there will be some loss. But that's it. Nothing for the beneficial effects coming in. So yeah, I don't think there's any such click and get something. I mean, I would love for it, because it is so hard for me to think of all of it. Head hurts. But yeah. All right, good. I think I'm done with what I had in mind. If there are no more questions, we can stop here and come back at 2. Okay. So 2 p.m. we have a research seminar by Himani Sachdeva. So see you all there. Bye.